DNA sequencing involves the use of dideoxyribonucleic acids in order for us to determine what the sequence is of nucleotides in an unknown sequence of DNA. And to begin with, the first place to start is by analyzing the structure, the chemical structure of the sugars involved in RNA and DNA, as well as another kind of sugar called dideoxyribose. And so in RNA, we have the typical 5-member ring. We have the 1' carbon here. We have a hydroxy group on the 2' carbon, as well as the 3' carbon. And then we have an additional carbon here, and the base would be attached to this. So the identity of whatever RNA it is, AUGC, is determined by whatever this base is here. And this is ribose. And so this is what's found in RNA. Now, in the case of DNA, this 2' hydroxy group gets turned into just another hydrogen. And so in deoxyribose, in deoxyribose, this deoxy part refers to this 2' hydroxy group being nothing more than a hydrogen. And in the case of dideoxyribose, what that means is this 3' hydroxy group will now also be nothing more than another hydrogen. And the consequence of this is when a DDNTP, a dideoxyribonucleic acid, is added onto a nascent strand of DNA, this 3' hydroxy group that was previously used to link to the phosphate group on the 5' carbon can no longer do that. And therefore, the growing strand will stop growing. And so I'll go over an example. Let's say we had sequence ATGC, and we had DDCTP present in our solution. And so DNA polymerase would be here adding on the complementary base pairs in the 5' to 3' direction. So it would correctly bind thymine to adenine, adenine to thymine. But when it adds on this DDCTP, we stop sequencing everything after that. And so what we would have in our solution is a fragment we would have the complementary strand, we would have TAC, and that would be it. And so we would have, of course, present also DNTPs. So if our sequence had another G, for instance, it would be more likely that DNA polymerase would add the correct DCTP to this G, and we could continue sequencing. But what we get out is a bunch of fragments such as this. We'd have the TAC if DNA polymerase added in the DDCTP at this G. We would also have a TACGC due to DNA polymerase adding on the DDCTP at this G in our sequence. And so we would have many fragments present in our solution. And this won't just be caused by, we wouldn't just have DDCTP present, we would also have DDATP, TTP, and GTP. And so we would have many different fragments, and for each one they would end in whatever NTP we had present in our solution. And so the breakthrough with Sanger sequencing that allowed us to even be able to figure out what DNA sequences are in the first place is this. So if we take these fragments that we have and we stick them into a gel, and we would be able to isolate the individual fragments that corresponded to each dideoxy nucleic acid. In our well, we would have lanes that would correspond to different fragments. And so the shortest fragments would get through first, and so our ladder would look like this. And so if we look at which fragments of DNA travel the farthest, that means they are the smallest. Whichever ones got through the quickest would be tell us the order that the DNA base pairs made it through. So this would be 1, 2, 3, 4, 5, 6, 7, 8, 9. And so that tells us, because the lowest and the smallest DNA fragment that made it through was had a guanine in it, it tells us that the first nucleic acid in the 5' direction that we were interested in is G. Because the second one was a C, it tells us it's a C. Because the third one was A, we know A, T, and we can continue this on and derive what the DNA sequence is of the unknown DNA. And this was a huge breakthrough. The key restriction, though, is we are limited in the size of the number of base pairs that we can sequence with Sanger sequencing. And the problem arises when we have very long DNA segments, because once you get to 1,000 base pairs or thereabouts, the difference in mass and size between a DNA fragment that is 1,000 base pairs and a DNA fragment that is 1,001 base pairs is so small that it becomes negligible. And consequently, if we wanted to sequence something like the human genome, which has 3 billion base pairs, what we need to do with Sanger sequencing is turn to something called, shotgun sequencing, and that relies on us breaking DNA fragments into things smaller than 1,000 base pairs and then sequencing those. The problem with that is that you now have a very complicated math problem or a letter problem, and you have to figure out where all of these sequencings overlap with each other in order to determine what the actual DNA sequence is for anything larger than 1,000 base pairs. And so this concludes Sanger sequencing. It's also referred to as shotgun sequencing often in textbooks and classes. But I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.